Morning. We'll begin with lecture 33 today. We begin our discussion of the development of the nervous system with the study of the development of the spinal cord. For an overview, we recall that the entire central nervous system and peripheral nervous system is derived from the neural plate, the ectoderm, and that is induced by the notochord that had originated from the pit and some of the paraxial mesoderm. We have looked at before, and we'll review again quickly, the development of the neural tube for the CNS and the neural crest giving origin to the peripheral nervous system, at least the sensory and postganglionic motor autonomic neurons of the autonomic nervous system, both parasympathetic and sympathetic postganglionic neurons. The remainder of the neural components of the PNS, peripheral nervous system, including the autonomic nervous system, of course, are axonal projections from the neural tube, the central nervous system itself. So the neurulation begins early in week 4. We recall that that was a very early event, just after the formation of the embryo in the 3rd week. The cranial to the 4th pair of somites are the landmarks to where the brain, the rostral 2/3 of the neural tube, is going to form. And that's a complex array of events that will be discussed in the subsequent lectures. The caudal 4 pairs of the somites contribute or are the landmarks for the area of development of the spinal cord, the remaining 1/3, caudal 1/3 of the neural tube. We recall that there ultimately was a closure of the neural folds to form the tube, and it progressed basically from the midpoint rostrocaudal as the origin and extended rostrally and caudally from that central location, always having some opening at both ends, at each end. And there will be closure of both of those rostral and caudal neuropores at some point. This closure being referred to here, I jumped the gun a little bit, is really talking about the fusion of the folds to close the plate into a tube. But that's fine. We know that accompanying that closure is the sequential closure, getting closer and closer to the final closure of the pore created by this rostral and caudal closure of the folds. And there is open communication with the amniotic cavity as with the digestive system as well. So the rostral neuropore is what is generated, and it proceeds rostrally as these folds do close. So it's a relative point that migrates rostrally with this closure of the tube, of the folds to form the tube. And it ultimately will close with the final closure of the neural folds rostrally on about day 25. So again, this is 4 days into the 4th week. Day 21 is the end of the 3rd week. The caudal development of the embryo and fetus, as we know, is delayed by a few days, and it's represented here by 2 full days for the closure of the caudal neuropore. And again, we see the angiogenesis is a triggering factor. So we recall that angiogenesis has contributed to the development of many structures. The most recent discussion was with the skeletal system. The neural canal that forms the central part of the neural tube will give rise to the ventricular system, the cerebroventricular system, lateral ventricles, 3rd ventricle, the interventricular foramen, the 4th ventricle, and the connection between the 3rd and the 4th ventricle. Not remembering that term right now. Trying to rush my way past it. And of course, the central canal of the spinal cord. So this brings us way back to this early view of the developing embryo We recognize the primitive pit right in the center and the notochord extending cranially from that to the prochordal plate, prechordal plate, oropharyngeal membrane, buccopharyngeal membrane. One little spot with 4 names, and remember that was our landmark very early on. The notochord does not extend any further forward than that. This notochord triggers the differentiation of the overlying, along with the paraxial mesoderm that's developing simultaneously, although very early at this stage, contributes to the differentiation of the overlying ectodermal cells to form first the plate, really a heightening increase in the height of these columnar cells. And we're going to take a look at a cross-section here to remind ourselves of that. Extending caudally is the primitive streak in the cloacal membrane. This looks as long as the notochord up to the prochordal plate at this stage, but this area up here is going to grow very rapidly, and relatively this becomes extremely small, and I almost said inconsequential, but we know nothing is inconsequential in the embryonic development, but relatively negligible as far as dimensions are concerned ultimately. Thus, we are reminded of this image, the cross-section, with the endoderm, mesoderm, and ectoderm, and the notochord with the early paraxial mesoderm stimulating the overlying surface ectoderm to increase in the height of the individual columnar cells, simple columnar cells, so that there is less height in the very central midline running from rostral to caudal, at least just overlying the notochord, forming the neural groove, and the taller cells on each side forming the folds. And that collectively becomes the neural plate that undergoes a tremendous amount of change with the size and the folding, as we recall, with the rapid growth of the embryo. To reflect some of the changes that occur throughout the length of the embryo at this stage, we can see there are many somites present at this time. We start at the level D near the rostral neuropore and go more centrally located at level E and level F for these subsequent panels. And basically we'll see this again and again. It's just a progression of the change in the overlying ectodermal cells so that the crest becomes more enhanced, and as well the neural groove becomes accentuated until these crests become the folds and fuse, ultimately meet the contralateral side and form the fusion. The paraxial mesoderm is contributing to the somite here, as we recall. As we go more caudally, it's more developed. As we get closer to the origin of the notochord, these are our friendly landmarks, notochord dorsally, and eventually we transition over to the neural tube, as we can almost call it here, as there's almost a closure of the neural folds. The cells just at the junction of the point of fusion of the neural folds becomes very important at this neural crest, because the neural crest cells are going to form from that area. Farther caudally, we see that this has already occurred. So many of these cells that had been part of this layer up here have now detached, and they have also detached from the neural tube. The neural folds would have fused here dorsally. So these neural crest cells are going to be very important for the development of the peripheral nervous system, not just the neural component, with the exception, of course, of the axons that ultimately come from the developing spinal cord, but also for all the cells that surround any of the neural components, in particular, of course, the Schwann cells, but there are some other supportive cells that surround the sensory neuron cell bodies called satellite cells. I'm not trying to implicate that the connective tissue and vasculature of the peripheral nervous system originate from these. We know that those are mesodermal origin. So again, to this view of the 23 days, there are several somites. The neural tube is quite well formed. We see the rostral neuropore and the rostralmost neural fold with the groove very accentuated as there's a rapid growth in the development of the neural primordial cells that will ultimately contribute to the formation of the brain of the central nervous system. So we're beginning to recognize already the brain, brainstem, and spinal cord here, and we see the caudal neural pore as well. The rostral neural pore is approaching closure more rapidly than the caudal neural pore, as we recall. From this lateral view, we can see that there's a tremendous forebrain prominence. We know that the brain becomes quite large compared to the remainder of the central nervous system, so this is not shocking. And the rostral neural pore is now closing in this instance. This is a mid-sagittal view, lateral view of a similar embryo, and we can see at this point the rostral neural pore is open, and it is continuous with the amniotic fluid that we see here. This comes all the way around to this structure that we recognize as the oropharyngeal membrane. It was that original prochordal plate that was out here. The rostral fold has now pulled this down in. The notochord did not extend beyond that point, and the nervous system is developing at all locations where we find the notochord. And we notice now that it appears that the notochord extends farther caudally than what it may have originally. And this is what I was talking about in that previous slide, where the primitive pit back to the cloacal membrane becomes negligible once we see the relatively rapid, much more rapid growth rostral to that, and this is what I was referring to. The tail fold is also occurring here, and so we see now the neural tube extends all the way from the caudalmost portion of the embryo to the rostralmost portion, and we're going to follow the development of the spinal cord, that caudal 1/3 of the neural tube, to see the closure of the caudal neural pore and the cytological development of the spinal cord itself throughout this entire region. So the progression, as we noted, is neural plate, neural groove, and neural tube. And we can see that in this series of cross-sections of an embryo, and it's from the same embryo, just progressing from caudal to rostral. Here, the caudalmost portion here is the most highly developed. We see the paraxial mesoderm, the extra-embryonic coelom developing there. The crest is obvious here, so some of these cells are the primordia of the neural crest cells. Then we see really just the plates with the fold, and it is not quite forming this clear crest, kind of like a wave with the crest of the wave. These two obviously will join together. And as we progress rostrally from the origin of the neural plate, we get just back just to the point where the neural plate is there with the neural fold. Then at 47 days, we can see the full length of the neural tube on this 3D reconstruction from an MRI of a human embryo. We see all of the somites, so we know the paraxial mesoderm is there, and the rostral neural pore has not quite closed at this point. But let's now begin to focus on the development of the spinal cord in the peripheral nervous system. This is a scanning electron micrograph of a cross-section of a mouse embryo showing the neural tube. We see that it has fused and closed off dorsally, and some of the cells at that point of fusion have taken this position dorsolateral to the neural tube. We notice the notochord here located ventrally. This would be they have an S here for somite, but that would be the paraxial mesoderm. So the neural crest cells are beginning to migrate into this area. They are artificially colored in red in this panel on the right-hand side, and we see that they are indeed beginning to migrate away from their point of origin where the neural crests fused to close off the neural tube. So we'll follow those in a little while, but we know that what they contribute to is the formation of the peripheral nervous system. For now, let's just step back from that overview and consider the development of the spinal cord itself. Then we'll go on to the peripheral nervous system. So the lateral walls of the primordial spinal cord grow, and they are composed of pseudostratified columnar epithelium. This means that it's still a single layer of cells, but some are really tall, some are squashed in between, but they all have an attachment to the ventricular zone very close to the neural canal that had been formed. So then we are considering a ventricular zone close to the neural canal and a marginal zone on the periphery surrounding the outside of the neural tube. So this ventricular zone is where we see a tremendous amount of mitotic activity, as we'll see in illustrations in a moment. So all the neurons originate from that mitotic activity. The astrocytes, oligos, and ultimately a layer of cells remains called the ependymal cells that we're all quite familiar with. They have cilia, and we know that the cilia project into the cerebrospinal fluid within the central canal of the spinal cord or the neural canal of the neural tube at this stage, and those cilia move that cerebrospinal fluid. The ependymal cells also contribute to other structures, as we'll see in the future slides here. The marginal zone is at the edge, gives rise to the white matter, and then there's an intermediate zone as well, and this will give rise to the gray matter. So ultimately, ventricular and marginal zones give way to a three-layered spinal cord: ventricular, intermediate, and marginal. And this is what we're going to look at here. Note the coloration: the darker blue on the ventral portion of the neural tube and the lighter blue on the dorsal. There is a sulcus that separates these two called the sulcus limitans. We'll see that in another slide. And that sulcus limitans becomes very important because, as we will see, everything dorsal to this becomes committed to sensory processing, and everything ventral to that sulcus limitans becomes dedicated to motor processing. So dorsal sensory, ventral motor. The neural crest cells are laying down the primordium of the spinal ganglia. And of course, this dorsalmost portion represents the site of fusion of the neural crests at the edge of the neural folds. When we jump ahead to another stage of development, we can see that the marginal zone is becoming accentuated. And what we need to remind ourselves is that even though we are seeing the sensory neuron cell bodies and their central processes and peripheral processes that had developed from the neural crest cells, and the motor cells, the motor neurons within the ventral portion of the intermediate zone, and their axons extending into the peripheral nervous system. There are many other neurons throughout here in this alar plate or the dorsal zone, also from higher centers. So there are many axons that have been sent out into this marginal zone to form circuits that occur between different levels of the spinal cord, between the spinal cord and the brain stem, the spinal cord and the thalamus, as well as many descending axons that are coming from higher centers: brain stem, motor centers, and sensory integration centers, and pre-motor autonomic nervous system neurons all through the brain stem, and of course any cortical neurons and anything from the cerebellum or other structures. All these axons are coming into this marginal zone and trying to locate their target neurons to form the relevant synaptic circuitry essential to, of course, coordinating sensory and motor processes. So even though they're just showing this pale blue coloration, we know that it's loaded with axons, most of which become heavily myelinated, and we'll talk about the development of the oligodendroglial cells that myelinate axons in this area. But there are some unmyelinated axons out here, not many, but within the gray matter that comes from the intermediate zone, there will be a tremendous amount of unmyelinated axons. And we remind ourselves that even unmyelinated axons are surrounded by, not wrapped, but they're surrounded by the oligodendroglial cells. What we're seeing then in this slide is the prominence of the sulcus limitans, and as we get the distinctive development of the ventricular zone, this is mostly ependymal cells by this point, and the dorsal portion separated from the ventral portion, that there's actually plates formed of cells in the dorsal part of the intermediate zone and the ventral part of the intermediate zone, and they become alar plates. Alar means wing, and basal plate, obviously, at the base or the ventralmost portion of the intermediate zone of the developing neural tube. The marginal zone still is out here, as we had stated, becoming established as the white area of the spinal cord, white matter. So there's a roof plate that closes off the neural canal, going to become the central canal of the spinal cord. This is the site of fusion of the neural folds, and the floor plate, the original deepest part of the depression of the neural groove. Notice the term neuroblasts, so motor neuroblasts and sensory neuroblasts. So these, anytime we see blast, we know that these are early cells. As we proceed through the development, we can see that the intermediate zone of the neural tube, now we need to just call it the gray matter of the spinal cord, becomes much more highly developed. The ventral horn, committed to motor function, becomes very prominent. There's an intermediate zone that, at least in the thoracolumbar region, T1 to L2, and in the sacral region, S2, 3, and 4, becomes committed to the visceral motor neuron component, the preganglionic autonomic neurons. But everything else is committed to sensory integration, and many of them are interneurons. These neurons just are not shown in this area, but we can see the axonal projections of the motor neurons. It would even be the visceral motor neurons that would be sending out their axonal projections, the preganglionic sympathetic and parasympathetic neurons, depending upon where you are in the spinal cord. So these are those peripheral projections of these central neurons that we said end up in the peripheral nervous system, but all the peripheral nervous system that is related to cell bodies within the peripheral nervous system originate from the neural crest cells as these sensory neurons in the dorsal primary and in the dorsal root ganglion, of the spinal cord. So we see the marginal zone has become much more highly developed, many more myelinated axons, and all the other related cells, the glial cells, to complete this. And so it's become a tremendous extension from that original roof plate. So this dorsal portion of the marginal zone becomes very prominent, as do the other areas, and actually forms a septum far from the original dorsal roof. So this just represents the amount of axonal growth into this dorsal column area and the heavy myelination by the glial cells that are there as well. Although it doesn't seem as prominent, it actually is as dramatic for the lateral and the ventral white matter of the spinal cord. The ventral portion, there is the anterior spinal artery, so we don't get this fusion of the two anterior or ventral columns as we see dorsally. Again, structures develop around and in relationship to pre-existing structures. So if there's an artery here, it doesn't fuse together completely. We can now go to this histological sample. and see that there is a dark staining of the gray matter. So there are a tremendous number of neuron cell bodies and their dendrites, as well as a lot of unmyelinated axons in this area. But we can see distinctively the sulcus limitans, the the ventricular zone of the neural tube, mostly ependymal cells by this point. We can see the alar plate and the basal plate and the marginal zone beginning to develop. Here's the roof plate and the floor plate. We also see the spinal ganglion, the dorsal root ganglion. So any neuron cell body and its surrounding protective cells, the satellite cells in here, originated from neural crest. We can't really see the axons, the motor axons coming out, but they are coming out here to join the peripheral processes of these sensory neurons, to form the mixed spinal nerve. Developing around it, we can actually see some of the cartilaginous molding of the vertebra that is going to encase this ultimately, as we know. Thus, we come to this point of looking at the high magnification illustration to look at some of the cytogenesis of the spinal cord. And first we note the neural canal would be to the left, and the area adjacent to the developing vertebra would be to the right. So we know there's mesenchyme here and amniotic fluid over here. This is at the point where there's already a neural tube, so we're at a point where the folds had closed. So there is a protective thin membrane there called the internal limiting membrane, and we begin to see that there's what appears to be multi-layered cells, but it actually is pseudostratified. All of these cells, somewhere along the way, this looks like it's not attached, but maybe in the section back closer to me or something, this portion of this neuroepithelial cell will still attach to the internal limiting membrane. So all these cells have a cytoplasmic attachment to the internal limiting membrane. Some of them reach the external limiting membrane, but not all of them. So this really is just one layer of cells by definition, because they all have an attachment to the internal limiting membrane. But it looks like it is multiple cells, layers thick, therefore the pseudo-stratified epithelial terminology. So these neuroepithelial cells are undergoing tremendous growth as we move away from the ventricular zone. When we look at the ventricular zone, we can see this is the area of a high level of mitotic activity. Even as we proceed through the stages of development, the dividing neuroepithelial cells will be most prominent at the ventricular zone. We see now an establishment of the intermediate zone. So some of the cells now have been pushed away, if you will, and no longer will attach to the internal limiting membrane. And we see the marginal zone out here. So we know this intermediate mantle or zone is going to be where we will see a lot of neuronal cells develop. And obviously many of the cells out here are going to become committed to becoming glial cells, because they're going to have to make an association with the axons that invade this area, if you allow that terminology. And obviously the mesenchyme that was on the outermost portion, the periphery of the neural tube, is going to differentiate into the three layers of the spinal meninges, the pia, arachnoid, and dura mater, respectively, from the surface to the outermost portion. So let's follow some of the cytodifferentiation that's going to occur. So what we had looked at was a cross-section of the neural tube. We saw the neuroepithelium and its differentiation into the ventricular, intermediate, and marginal zones. And the different cells will follow different lineages. There is the one exception, we'll point that out now. From the mesenchyme comes the mesenchymal cell line that will differentiate into the microglia. So the microglia is the one exception to the glial cells in that it does not originate from neuroepithelial cells. All the other glial cells originate from the neuroepithelium, as well as the neurons. So we see that different neuroepithelial cells undergo a lineage. Some will become committed to become ependymal cells, obviously those that stay closest to the ventricular zone. Others will differentiate down the lineage to become a glioblast, or it's also called a spongioblast. And there is an apolar neuroblast. So there is a cytological differentiation or distinction to each of these cell lineages, and we'll continue on to follow their development. So the apolar neuroblast will begin to extend its cytoplasm in opposite poles and become a bipolar neuroblast, but it will not stay that way for long, as we will see in the next slide. This multipotent, or let's just say bipotent, glioblast or spongioblast can differentiate along two lineages, as an astroblast or as an oligodendroblast. The ependymal cells obviously stay where they are, or they become associated with invading vasculature. In this case here, we're picturing them lining the entire cerebroventricular system, the central canal, and the entire ventricular system of the forebrain. As well, anywhere that there is choroid plexus, ependymal cells will overlie the encased or encapsulated vasculature. Let's return then to this lineage of the bipolar neuroblast, the astroblast, and the oligodendroblast. The neuroblast lineage we see again starting from the bipolar neuroblast. It becomes a unipolar neuroblast. Remember, this is the central nervous system only. And then ultimately this one process will become the axon, and then later there will be cytoplasmic extensions to form the dendrites. And we remember that there's bipolar neurons, well, not in the central nervous system. There's really just going to be multipolar neurons, but the multipolar neurons have a wide variety and morphology of the dendritic origins and branches. The astroblast is bipotent as well, can differentiate either into the protoplasmic astrocyte or the fibrous astrocyte. We know that some astroblasts do persist into adulthood. If there's damage to the nervous system, there will be an astrocyte scarring, astrocytosis. We now know that some neuroblast cells persist into adulthood as well. They're very limited in location, but this is a novel understanding of the nervous system related to its plasticity. The oligodendroblast, obviously, a single lineage become an oligodendrocyte. The oligodendrocytes will myelinate axons in the central nervous system, and as well, any axon that is unmyelinated is still going to have a process of an oligodendroglial cell adjacent to it. It just doesn't form the concentric rings. Thus, the developing cord differentiates into the dorsal and the alar sensory plate and the ventral or basal motor plate. So dorsal sensory, ventral motor. The dorsal root ganglion develops from the neural crest cells, and these develop into bipolar cells, the same as what we had seen, but then it doesn't lose the second process, it just fuses them together, so it looks like a unipolar cell. It's called a pseudo-unipolar, and we see that in this illustration. So here's a neural crest cell, looks just like that apolar neuroblast, but it originates from a neural crest cell, not from a neuroepithelial cell. It becomes bipolar, and rather than losing one of the processes, there's just a cytoplasmic alteration of the positioning of these two processes until they are basically one that extends from the soma and later bifurcates to form the peripheral process going out to the sensory receptor and the central process that goes into the central nervous system. So the meninges form from the mesenchyme surrounding it. The CSF will begin to form during week 5. So this means that both the rostral and caudal neuropores have closed, and therefore there's no more amniotic fluid flowing through this neural canal. So we have to have the choroid plexus formed. So the plasma seeps through the ependymal cells lining it to form the cerebrospinal fluid that is contained within the central canal, the spinal cord, and of course the cerebroventricular system of the forebrain and brainstem. The primordial spinal cord extends throughout the entire vertebral canal as dura and the vertebral column grow. The spinal cord appears to migrate, but actually it's just staying where it ended originally, and everything else is growing away from it. And we'll look at that in an illustration in a moment. The myelination begins late in the fetal stage, so even though the oligos get there, they don't form this heavy myelination. They form their intercellular associations with the axons, but they don't complete that multiple wrapping until the late fetal stages and even through childhood. So this is an ongoing process, that myelination. So as motor skills begin to develop, the final development of the myelination proceeds with it. So here on the left in panel A, we can see at 8 weeks the spinal cord extends all the way to the tip of the vertebral column, attaching to the coccyx bones. And then here in panel B, we see at week 24 weeks it no longer extends all the way to the tip of the coccyx, although the meninges do, but the actual neural component does not. Here in panel C, we see the newborn, and it's almost at its adult position, somewhere at the first or second lumbar vertebra, as we see here shown in the adult. The meninges continue down to attach here most caudally, and that's described in more detail in another recorded lecture, and I won't take the time to do that because I want to discuss the malformation of the spinal cord after we cover this one slide here on the cauda equina. So if you start with a spinal cord that extends to the full length of the vertebral column, and you have the segmental origin of the peripheral nerves, then when that portion of the vertebral column grows away from the end of the spinal cord, you just have the extension of the fibers in the connections between the spinal nerve and the spinal cord. So they become these long, pronounced connections, and as you all might know, these are the dorsal and the ventral roots of the spinal nerves, and they extend down, and it looks like a horse's tail, and that's the cauda equina. So in the 3rd month, the spinal cord extends through the entire length of the embryo, as we had seen, all the way to the tip of the coccyx. As the age progresses, the column and the dura mater lengthen away from the tip of the spinal cord that had been the neural tube, and the caudal end shifts thereby to a higher level. It stays where it was, but the vertebra move away from it, and as a result, the dorsal and ventral rami of the spinal nerves, or roots, run obliquely to their segment of the origin corresponding at the corresponding level of the vertebral column. So they're just stretched out. Let's now spend the last few minutes talking about spinal cord defects. There's just a few of these that we consider. So most common ones are related to failure of closure of the neuropores during the 4th week. If there is a failure of closure of the caudal neuropore, and there can be varying degrees of this, the clinical term is spina bifida. And the spina bifida specifically is talking about non-fusion of the vertebral arches, but it may include complications of the spinal cord itself. So it has a wide range of variations. This is typically associated with a deficiency of folic acid or an excess of vitamin A. So the prenatal care with nutritional education is extremely important. Failure of closure of the rostral neuropore is associated with this as well. There are other environmental factors, for example, hypothermia, that can disrupt the development at these stages as well. So here we see some examples, panel A and panel B. These are sketches showing the variations of spina bifida and the typical anomalies that would be associated with them, involving the vertebral arch, the spinal cord, and the meninges. A is the spina bifida occulta. So the spinal cord, the caudal neuropore, has closed completely in this, so there's no complication of the spinal cord itself. The meninges are completely formed and encased within the vertebral canal, but the neural arch does not fuse completely. So we recall that that final tip of cartilage was way back here where the neural arch would be closed and the spine of the spinous process would have formed. Well, somehow that didn't happen in this case. And in the overlying surface ectoderm, there's actually a mole that forms and grows coarse, dark, thick hair. So this is a typical sign of spina bifida occulta. It is hidden. This can extend from that degree to one where there's a complete failure of formation of the neural arch, and the meninges with the cerebrospinal fluid within it actually form a bulge on the surface, but the spinal cord itself has closed, the caudal neuropore has closed, and this is the spina bifida with a meningeocele. So it is a seal or a space, a cavity or cyst, if you will, formed by the meninges. This can extend all the way to a meningeomyocele, where the spinal cord itself will be contained in that cyst. And obviously, so far, everything can be surgically repaired. There's no major problem with this. Obviously, in the case in panel D, where it is spina bifida with myeloschisis, a schism is a split between things. So myeloschisis, the myelo refers to the spinal cord. This is a split of the spinal cord. Actually, it's not a split, it's just a failure of closure of the caudal neuropore. And we actually still have the neural plate and the neural groove, and the neural crest had formed, but it just did not fold up and fuse in the midline. So we not only have the failure of the formation of the vertebral arch, the ectoderm, the surface ectoderm forming the skin, and the edge of the neural crest, the edge of the neural fold, are still contiguous, and the actual, what would be the equivalent of the ventricular layer, this would be the ependyma on the surface here, is actually exposed to the external environment. This is a severe case and is extremely difficult to repair. We now take a look at a case. This is case 18-2 from our text. A male infant was born with a large lumbar meningeomyelocele. So that's this case here in panel C. Covered with a thin membranous sac. Within a few days, the sac ulcerated and began to leak. A marked neurological deficit was detected inferior to the level of the sac. What is the embryological basis of this anomaly? What is the basis of the neurological defect, and what structures would likely be affected? A neurological defect is associated with meningeomyelocele because the spinal cord and or nerve roots are often incorporated into the wall of the protruding sac. This damages the nerves supplying the various structures, so there's actual physical damage to the nerves. Paralysis of the lower limbs often occurs, and there may be incontinence because this typically occurs in the lower spinal cord. Remember the S2, 3, 4 for the autonomic control of the urinary bladder. And for the rectum and anus. So there's paralysis of the sphincters, of the anus, and the urinary bladder. So this finishes our lecture on our discussion of the development of the spinal cord on lecture 33. I appreciate your time and attention, and I'll see you next time.